entertaining chai time, and to that end, we have a great panel of people lined up here to talk about. I'm just going to change the preview so you can see everybody. I have asked everyone to wear something festive. Hamley failed the festive test. He's in India, probably couldn't find a Christmas jumper. We forgive you for that. I think Karin might actually win because not only is she wearing a Christmas jumper, but if she points the camera to, she's coming in from Cambridge, Massachusetts, and she swivels the camera to the left. It's got real snow. So that is real winter. I didn't, you know. So I think she wins the points for festivity. So welcome, Karin. Peter made a great effort there. He has actually worn a Christmas tree underneath, but it's probably too cold in England and in his house, and therefore he's got a little Santa rotating thing. So good for that. So welcome, Peter. Are you in Oxford at the moment?
Yeah. It's pretty cold.
Welcome, Chris. Coming in very early in the morning from San Diego. So great effort to get a bit of Christmas spirit and joy into it with your tinsel. As it's 7 o'clock in the morning or so there. So welcome from San Diego. And welcome Hamley coming in from India after a full-on day in the office, project managing and managing a very large team of chemists there. And appreciate your time coming on later from Ahmedabad. So I'm going to quickly start by asking each of you to introduce yourself. If you're not talking, in the background, might be Karin. So I'm just going to ask you just to introduce yourselves, a little bit of your background. You're probably known to a lot of the people on our chai time today anyway. But then maybe you could say a few sentences about what you think makes a good drug hunter. So over to Karin first. So Karin, please introduce yourself.
Hi everyone, and Sunil, I needed to change my microphone. Can you hear me?
I can hear you.
Okay, great. Well, thanks for having me. I'm so excited to be here with this wonderful panel, and of course all of you who are tuning in today. What makes a great drug hunter? There are many ingredients, and let me just start with some general ones, like the passion and courage to really explore the unexplored, not what has already been done. And that takes the courage to also, think about what is a good project, where can we push the boundaries on the science, but at the same time, really thinking a good project is something where when we have delivered in, it's not just a small reward, but it's a really changing the medicine and the practice of medicine. It takes good science to really think about bringing the different disciplines together and not being afraid of having people with different opinions come together, and then really having a master plan to carrying it out well, and not being afraid of perhaps one might fail. I always say if we don't push the limits enough, if we never, that's a sign when you never fail, you haven't pushed the limits enough. But I really think bringing the different disciplines together and following a good plan on a project where we know when we have delivered it, we really want it, it makes a difference to the patient.
That's a fantastic introduction. Thank you for that, Karin. I'm going to go over to Hamley next. So Hamley, please introduce yourself and let us know what you think you need to do to be great drug hunters.
Yeah, first of all, Sunil, your hat is very nice, looking very nice. So thanks for having me on this chai time. Yeah, hello everyone. Myself, Emil. I work for o2h Discovery, is the head of chemistry. So I think I'm the only organic chemist in this panel, and probably a little bit different from what other expert panelists are there. In my opinion, the chemist role for drug discovery is to make the molecules they are assigned for. So I would like to say that how can a chemist be a drug hunter? Yeah, I mean, don't get down by the difficult molecules you have passed on from your collaborators to make. Stick on to that until and unless you find a route to make those molecules. Please don't give up. I mean, chemistry is always challenging, and we also understand that our collaborators need to change their designs, change their models, and change their priorities. But we need to keep working on them so that, I mean, ultimately we are working for a single goal, that is giving a good drug to the market, right? So as a chemist, I mean, even if it is midnight, please don't stop thinking about the ways in which you can crack your reaction or else find your molecule out. And yeah, I mean, enjoy the day when that molecule is made. So that's from my side.
Thanks, Hem. There's a lot of passion, energy, and focus on making the right molecules. We don't tell him to work till midnight, by the way. It's not, he may do that, but yeah, thanks for that, Hemel. Hemel has an amazing amount of passion and energy for what he does. So, Peter, over to you next.
Okay, thanks for inviting me, Sunil. I'm a chemist by trade, actually, Hemel. I started.
What he meant was he thinks he's the only synthetic chemist rather than medicinal chemist. I'm clarifying. I know you are all chemists.
I started at AstraZeneca and then I moved to Sanofi, and I was part of many drug discovery projects in the past. Then business development and head of external innovation for drug discovery technologies. And then I decided that it would be much more interesting to actually join one of these innovative early companies. And so now I'm CSO of Samsara, which is a small biotech. It's about 2 years old, and we're looking for inducers of autophagy for various different aging-related diseases. So what makes a good drug hunter? I think one thing that is a bit painful is experience, because it's an incredibly complex subject. And I think you need to really have seen quite a lot and been through the wars in order to be really effective. And that also means that you have to have a really open mind to learn from what doesn't work and what works, of course, to change your strategies and techniques as time goes by. And that's one thing that I've absolutely loved about my time in this industry, is the way that people have changed their views about how to do things over the years. And fashions come and go, but I think as time has gone on, we've got better and better at doing drug discovery. So that's probably the most important thing, as well as lots of creativity and hard work, obviously.
Thanks for that introduction, Peter. By the way, we're going over to Chris next, but I'm going to ask the audience. We have a very good number of people there, and I can see that we have some experts in the audience all over the place. I will ask you to please come in with questions, put them on the chat. I'll try and work through as many of them as I can. We have a great panel here today. It'd be a good chance to ask them what's on your mind. So on that note, over to Chris.
Hey, Sunil, thanks very much for the invitation, by the way. This is pretty exciting for me. You know, I'm out on the west coast of the US. As you can tell, I'm from the UK originally. You know, I started my career at Pfizer down in Sandwich in the Sex Health Group, then relocated to San Diego, and then I've been working in a mixture of large pharma companies, some VC constructed incubators, and some biotechs as well. You know, one of my interests is in fragment-based drug discovery, and maybe we'll get into that a little bit later. But in terms of, you know, some of the things I encourage people is to, you know, it's all about idea generation, or a lot of it is about idea generation. And then, you know, as people generate ideas, is really to develop a belief in those ideas. Because almost by definition, if it's a new idea, it's going to be disruptive. And then as it's disruptive, you're going to get pushback. And as you get pushback, you know, you can either step back with that, with it, and then essentially walk away from your idea, or you can sort of double down on it and really use your belief, essentially, which is your, you know, the foundation to, in order to have the courage and essentially to push back against that. And then you generate, and then you incubate, and then you bring this new idea out to fruition. And then it's when you get out to there, and then, you know, people start then picking it up and going, oh, that's a good idea, and it starts to develop. But the actual initiation, almost, as I said, by definition, is disruptive. And so believe deep in what you're doing, and push hard. And when you get pushback, you know, don't get upset. It's actually just feedback that you've actually got an idea.
Yeah, I mean, championing the idea, seeing it through, and let other people pick up, and you feel good about your idea being adopted. You know, and you know, you almost have to go out on a limb and take that risk that, okay, this may not work out because it's drug discovery, but actually to get it through and get it started, someone has to have that like insane belief for their idea and project. That's a really good intro to everybody on the panel. I've asked different questions to different people as we go along. Because I know that everyone has different strengths. Again, I'm going to ask the audience. We have almost 100 people live today. Please come in with your questions. Otherwise, I will pick on people. You know who you are. I know nearly all of you. So please come in with questions. I'm going to ask my first question. It's going to be around hit finding, and it's going to be to Peter and Karin to say, look, what technologies do you or have you used successfully for hit finding? You must see quite a bit coming on your plate, especially, well, both of you. I know that Peter's involved and Karin has a broad spectrum. It's about creating that early chemical matter, making that good start can really help the timelines of drug discovery. So what do you find most successful in creating hit matter, or is it different for different types of projects? So first I'm going to go to Karin, and then I'm going to ask Peter the same question.
Thanks, Sunil. And by the way, there are some questions in the chat for you. Hit finding, and I actually like to talk about hit generation even more than finding, because it's not just finding the hits that are there, it's actually really generating hits. And when it's such an important area, which I think over the years has also now really evolved enormously, as we are going to after very difficult to drug targets. And being in an environment where we want to do that across many, many disease areas and indications across many different classes of modalities, also that, we use, as you said, a variety of approaches. What has worked out really well, in one sense, especially when we really know which protein we want to go about and how central that protein is structure-based approaches. And I'm an absolute fan of structure-based approaches, and we have been really had, as many others, a lot of interaction on structure-based approaches. And I think now with the cryo-EM, really also understanding much more and the bigger picture, the interaction of proteins and how we disrupt protein interactions, that adds another aspect to it. Doing that early together with great computer-aided drug discovery and combining the two, that has been extremely successful for us. In addition to that, on the other side of this is DNA-encoded libraries. And I've been really impressed with thoughtful libraries, what we are able to get done there, and how many projects we have been able to get to starting points where it was difficult. I want to say it's not just numbers. I'm a true believer, as referred before, Namil, that synthetic chemistry is an important aspect. How our libraries look like, what they actually explore is important, because the starting points, the hits, if we have good properties in those, we have a much better chance to go beyond having a tool. In addition to these tools, we have there where we look at it as more straightforward. We're using virtual screens, which has become very routine now in some of our work, and that helps us start in some of the targets where we think we know already pretty well. And then also very importantly, working in a company where we're really going after first-in-class, high-hanging fruit is thinking thoughtfully about our libraries of cell-permeable compounds and really phenotypic screens where we might not yet know the target or exactly the mode of action, and we go after. The combination of those four, I understand not everybody can invest in them. One can also focus on one and get a lot done. But when we work across so many disease areas, then we need to have the combination of those to really get to the starting points. Of course, the biological assays that help us then understand what we have are an important factor to success too.
Thank you,
but
Karin.
The chemistry and the synthesis in the library, what we have, it's extremely crucial.
That's where Hamley and his team kick in. Peter, same question to you, maybe with more emphasis on your experience around various technologies and how you can use technologies to support hit find?
Sure. So I think Karin has covered a lot of the things that are really important. But one of the things I wanted to mention is that I think over the years, the whole sort of target space has got more and more difficult for drug discovery because the so-called low-hanging fruit have usually been found. So in general, not always, but in general, if you give a drug discoverer a GPCR or a kinase, I think we've got really good techniques to do that. And as Karin said, if you can involve structure-based approaches really early, then there are lots of great examples of people doing great work there. One thing that we really specialized in Frankfurt during my time there was, I think, using parallel synthesis and libraries in an intelligent way so that we could really make really fast progress on some of our projects. But I think as time goes on, those low-hanging fruit get picked, and we start to look at more difficult targets. So we know that, you know, we want to modulate transcription factors, or we want to find protein-protein interaction inhibitors. But we also know that they're incredibly difficult, and standard techniques such as high-throughput screening start not to work. And we did a study in Sanofi. I won't give you the total statistics, but we did a lot of phenotypic screenings over a 10-year period. And sorry, not phenotypic, PPI screenings over a 10-year period, and really got almost no success in the end. So we have to start changing our technologies. And there are some really interesting novel plays out there from you know, small innovative biotechs. But the most obvious one, and I agree with Karin, that I think is starting to be routinely used now is DNA encoded libraries. They really seem to be working for tough targets. And we had a couple of amazing successes actually with Dell recently in Sanofi on targets that proved completely intractable to the normal methods. So that really convinced me that Dell is the way to go. And
have you tried any sort of AI-based tools, you know, like the Farm Enables of the world, the turbines, that kind of thing? Tried any?
Well, I think when you talk about AI, you have to separate the two things. AI to come up with new biology and new targets, which is extremely exciting, and AI, which is what Turbine is doing very well. But there's also AI to guide chemistry. And there, I think it's a little bit, I think it's probably a little bit more in its infancy there. There are great companies out there, including in the UK with Exscientia, one of the world leaders of using AI for compound design. So I think that will definitely pick up in the next few years, and we will see. But the problem still remains, for difficult targets, we need new technologies to come up with that. The last point I'd make is about phenotypic screening, because that's a shortcut to getting really interesting new biology. The problem with phenotypic screening is you've got to do it properly. You've got to have really translatable assays, and you've got to really work very, very hard to identify targets and mechanisms of action, which is what we're doing in Samsara.
With the phenotypic, I'm sure we'll comment on this later, but with the phenotypic screening, you really need to have that good depth of backup of biology, which I know that you would have in the big pharma to try to work out what the actual target is and backfill.
Yeah,
just been a challenge for some of the smaller biotechs, I think. I'm just going to go to Chris on this point of finding new chemical matter screening start points. He mentioned about fragment-based drug design. So, I mean, Chris, maybe you want to comment on how you think this kind of technology is supporting the biotechs mainly, and whether you think it is a good way to start a new project.
Yeah, so, you know, I've been involved with it now for a good number of years, and some of it's actually just totally out of necessity. So if you're a small company, you don't have huge amounts of resources, the actually cost input into a screen and potentially generating matter or lead matter. And also, I think, you know, there was a paper by AZ where they were, and maybe even they incorporate it on a regular basis of actually doing a fragment screen, seeing what the hit rate, and using that as an accessibility of, or assessment of druggability. But yeah, I've used it a lot, part of it is a lot of libraries. There's a lot of good libraries out there. You can build your own and get very sophisticated with that, like Aztecs. But essentially, an access to anywhere between 1,000 to 2,000 compounds is very doable. It's very manageable. I've even played it out a library myself, you know, with the pipette and all that sort of stuff. So if you're in a small biotech and you really need to get something done, it's very, say, accessible. But then also over the last 10, 15 years, the technology that's been now applied to the issue of sensitivity and being able to rule out false positives has moved on tremendously. And, you know, NMR as a technology, I think, was really big. I think in the early 2000s, it sort of disappeared a little bit, and now it's really come back again. And there's a lot of development in the NMR space. You know, one of the challenges I find is I really don't understand a lot of the NMR stuff that goes on. I use it, but there's an awful lot of physics, an awful lot of maths that goes behind the black curtain there. But yeah, I think it's, you know, it's definitely worth seriously considering looking for these.
We reviewed a lot of these AI or machine learning or quantum-driven technologies, and there are quite a few out there, and we've invested in a few as well. And ultimately, you've got to look at the examples and what comes out of them. So our due diligence is really hard for us to due diligence the long division, let's say. But what we can see is what we input into the model, what came out the other end, and actually did it generate something that looked like a drug or a good target or something. So these worked examples are really important for us in the scientific community to understand if we can use them as tools to speed up our discovery programs, essentially by having better quality targets or better quality start points. And speaking of speed, I mean, Hamel, maybe you can comment on some technologies that you're familiar with that can support with synthetic chemistry and try to drive more efficient ways of doing reactions or project managing your chemistry projects.
Sorry, I was on mute. So in regards to helping the drug discovery in terms of productivity by using technology, I think one of the platforms that we have built at o2h is the CITC. That's a project management platform where we are able to provide real-time updates to our collaborators, and it also helps in accelerating the productivity, because the knowledge across the platform is visible to different people who work on the project, and they are able to contribute on their own. In terms of increasing the productivity, in terms of reactions, I mean, there are several technologies like running parallel synthesizers, etc. Yeah, so using all of them, we are able to make a great deal of compounds in the labs. So also microwave robotics that we are using, and the SFC technology has increased our output for separating the enantiomers and helping the compounds with the desired stereochemistry. So this is the kind of technologies that we are using in chemistry to help the drug discovery.
Thanks, Hamel. I mean, just to let people know the acronym, CITC is, you know, o2h is Chemistry in the Cloud, which was engineered and developed by Shah and Nilesh and used by collaborators led by Hamel globally. But we also do adopt this technology, deep matter, which is kind of a sensor in a flask, and it allows us to more optimally understand when reactions occur. I think Hamel's piloting that in the labs as well in India. My next question is going to be a little bit around. Actually, you know what? I'm going to take something from the audience. Make sure I pick off some of these, because now we actually have some. First, I can answer the first question really easily, which is, where did you get that hat from, Sunil? So I can say my daughter gave it to me, and she created the scene behind me as well. She's now off school, so she needs things to do, and she's been keeping busy doing these things. Mark asked, what do the panelists feel are the most exciting areas of drug discovery to go for in the next 10 years with respect to therapeutic areas, technologies, or otherwise? So this is going to be a fast fire question. So whoever wants to pipe in can pipe in, but I'm going to go to Karin first. Most exciting areas, technologies, therapeutic areas. We'll probably just do Karin and Peter and Chris on this one.
And I know you want me to be fast, so let me say 3 things. I think, you know, all classes like transcription factors, and there are many different subclasses, a lot of opportunity there. thinking about mode of action, you know, having introduced degraders, and other types of interactions where we actually don't go to normally, where we have allosteric modulation, has so much opened up what we are doing. So in that respect, the technology to thinking also about interactomes, proteins in the context of another protein and how we can drug it, I think that will be really key in the future. And I think we should take on challenges like not just having the first cure for certain cancer, but let's cure breast cancer. So we will have modulators into some diseases, but we also need to bring some where we have had good starts home to really get to cures. And I think different mode of actions, how we get to really deep down on resistance, will help us there.
Thank you, Karin. Peter?
Okay, I would say small molecule modulators of RNA, I think is a big area that's got a lot of mileage in the next few years. Because we can apply all our techniques of small molecule drug discovery to a really new area. I'd say slightly biased, but I think the whole area of aging and longevity as a different way of thinking about biology brings a lot of different opportunities for new therapeutics, not just make you live longer, but in all sorts of different areas, including neurodegeneration. I had another one, I've forgotten what it was. So in interest of time, I obviously need
Chris, do you have an answer, or Emil, either? Okay, Chris.
Yeah, so in terms of therapeutic areas, you know, I think there's still a tremendous number of, you know, acutely really severe diseases out there. You know, if you think about ALS and, you know, the deterioration that happens in those patients over a rapid period of time, you know, as an opportunity for what we do, I couldn't imagine a better opportunity to impact a family and lives. And then also in oncology, you know, there's been a lot of success in oncology, but there's also a lot of devastation as well that cancer causes. And, you know, thinking through and designing molecules, maybe less so for single action, but actually incorporate in the design of combination therapies, you know, really early. And I think there's a question in the panel about coming to the panel from, you know, can we design in, you know, additional modalities or different mechanism of actions into the same molecule? Yeah, maybe that would be useful. And then in terms of technologies, you know, this whole idea of this, you know, the degraders, I think is very interesting. You know, when you look at the PROTACs, you know, very large molecules, maybe they're sort of compartment selective, i.e. not really get into the CNS and areas like that very well, but you know, bringing some of these new modalities, well, actually good CNS activity, I think could be very transformational.
Thanks, Chris. My next question is going to be really slightly organizational. It answers one of the two of the questions actually in the chat as well. Is that some of us work in large organizations, some of us sort of from large to small. You know, Chris's company is probably the most unknown largest company I've ever heard of or seen. You know, he works for a company called Marathi, and they've got a valuation of 12 billion, and I'm like, really? But anyway, we'll come to it in a second. So you know, managing large teams is a conversation I often have with Karin actually and Hamley, because you know Hamley is managing a significant number of chemists and a huge number of project leaders and scientists. And so what do you feel are the techniques or your responsibilities as a leader in managing that large team, and how do you get the most out of your scientific base? So I'm going to go to Hamley first.
Yeah, I mean, when we are working with large group of people, in my experience, I've observed the most important thing, what matters is communication, because it opens a room for discussions. I mean, people may have different difficulties at different point of times. They may have different difficulties regarding their different projects. They may have chemistry issues. They may have some work related issues. They may have some family issues, etc. So in my experience, I mean, communication is something which I have found very useful. If we are able to talk with each other, if there are forums where people can come and share their issues and problems, then we are able to resolve them quickly. in terms of scientific inputs, I think having an opinion from a large number of people helps to resolve the issue faster. So, I mean, we should have forums and processes which encourage people to share their problems, scientific problems, and have opinion and perspective of others, rather than just working themselves on their own problems and scratching their minds of how to solve them. So taking involvement of other people matters a lot when we are dealing with a lot of critical science and research. Yeah, I mean, process is also something which has helped me and others probably also. I mean, if our processes are neatly designed, and while designing the processes, if we take inputs from the real stakeholders, I mean, the people who work on the ground, and if we approach them, what helps them to increase their productivity or what makes their life easy, and design our processes accordingly, then I see that there is a better output. All in all, it's a very complex thing, but yeah, I mean, doing some bits here and there, we can solve the jigsaw. Yeah.
Thanks, Hemel. So Karin, you work in obviously the largest organization out of all of us and spent a good amount of your career in large pharma. How do you keep these, and it partly answers the question from Robert as well, how do you keep the interaction, the innovation fresh between the biologists and the chemists, and keep that discovery? Almost in a smaller biotech, we might kind of be forced to, we have to work in these small teams, which, you know, people tend to think are more innovative or not. How do you keep that fresh, the organization alive, constantly at the cutting edge of innovation in a very large organization like Novartis? And I know that Novartis do it very well, so I'm not saying that pharma are doing it badly. Novartis obviously have a huge reputation for innovation, but how do you keep it alive? How do you keep that energy there?
And Sunil, it's such an important question because it's something to work on every day. Ultimately, it comes down to the science culture. I think the science first, and then also breaking it out in really teams being in their project or project family home together, interdisciplinary. And what that means, you know, it's like, let's carve out a tennis court and say we are playing tennis, have clear goals, but broad goals, but then not micromanage the how. Let the teams go at it. We have an approach at Novartis where we get teams in to propose what they are working on, finding different cross-disciplinary partners. But then we are getting together and working across the disciplines. What is the plan? And what do you want to accomplish in the next 18 months? And so sometimes there is a danger when management can get too nervous and then one micromanages every step. That is suffocating creativity. At the same time, one can't just get started and not know where do we want to go. So having clear goals in broad strokes and then having those really capable people we have, unleash them, have the just do attitude, speak up culture, and reward the teams. It's really important. Drug discovery is a team sport, and we need to reward the teams. And then as leaders, we are catalysts. It's putting out, asking the key questions. That I think is really important to get to those creative teams that continue to push the limits, continue to get beyond what we just done. Otherwise, one has to be worried that one just does what one has done before to be safe. We want our people to feel safe to take on risks, to feel safe to be clear where they can really have their playing field, and we are not micromanaging how they are doing it.
Thank you, Karin. That's a great answer. So Peter, you were a part of a large organization, probably one of the largest, and you've probably gone to one of the smallest in the world. And, you know, you have to now fight for your funding, fight for your resources, and you're probably doing everything yourself. 90% of all activities is done within your brain or within 1 or 2 or 3 people. So how do you find going from, you know, very recently, so this is fresh, from going from Sanofi, multi-geographies, multi-sites, everything done, you know, experts all over the place, to really doing innovation by yourself or within a 2 or 3 people?
Yeah. I find it very liberating, I have to say. And then that's one of the reasons I wanted to leave Big Pharma, was because I don't want to say anything too controversial, but I felt that there were too many decision makers, too many layers of responsibility, too much conservatism, too much, as Karin just said, that obviously Novartis doesn't do, but too much micromanagement of scientists, that kills innovation immediately. We were all in Sanofi tired of our good projects being killed for strategic reasons, because different managers came in and had different ideas. And so we destroyed, over a long period, we destroyed a lot of value, I think, and that's really frustrating for scientists. So in a small company, of course, there are big challenges of resources, but on the other hand, it is incredibly liberating to be able to make a decision and act on it immediately and not in a few months. And I always use a little, very small example of being able to sign a CDA within an hour rather than a month. That's the difference between a small company and a big company. And it makes a big difference when those things are added up. So I think as long as you do it right, you can make huge progress very quickly in a small company. And that's one of the big drivers, I think, of innovation. On the other hand, the painful side is you're always concerned about your next funding round. Fundraising takes a lot of energy out of the company and is distracting. And of course, you can't do everything you need. So one final example I'll give is our struggles to put together a high quality compound collection, because I always assumed it was there for me. It was free. We had too many compounds. That was always our problem. But now we have to try to find high quality compound collections without paying too much. So that's a completely different ballgame. It's possible, and we did it, but it took a lot of effort. So, you know, there's swings and roundabouts, but I prefer.
I'm going to move on to my next question, because I can't believe how quickly the time's going. I'm going to extend this for another 15 minutes or so till the end of the hour. You know, we all rely now on doing some of our innovation externally, whether you're the biggest pharma fully integrated in the world, or you're the smallest biotech, and obviously you rely much more on external innovation at that point. Now, working well with your CRO collaborators is important, and it's quite good today. We're able to see from both sides of the fence how to make those collaborations more productive. So I'm just going to ask the question as to really, you know, how do you see external innovation? How do you support that piece of the jigsaw? And what do you feel that you do to try to make it more productive? Or what do you think people should be thinking about? Because you've all got a lot of experience in this space. So I'm going to go to Chris first, just to keep it interesting.
Yeah, so we've been working, you know, with a lot of, you know, external partners. Is this what you're asking? Sorry, I was.
Yeah. So really the question is, what do you think? What advice would you give to people listening who have external collaborators to make it more productive, a more productive relationship?
I think relationship is the key. Yeah. So, you know, finding ways to build personal connectivity with the people that you're working with. You know, I think it was easier in the past. We could go and visit them. They could come and visit you. You could have some nice face-to-face interactions. And, you know, like all things, the more you work on the relationships, essentially, I think the better the relationship will be. So there's the personal component, but then it's also, you know, clear communication, clear organization on your side, clear organization, communication on their side, you know, to avoid any sort of classical, you know, miswiring, you know, so then it can avoid any negativity, and then it allows you just to sort of build and continue with the positivity and the outcome and results. You know, one of the things, you know, that I've got a lot of respect for over the years of outsourcing groups is in a way, when you're asking an outsourcing group to do something, that's what they do all day, every day. And then they're in a competitive, essentially capital environment or capitalist environment. So they need to be successful and they need to be good at what they're doing. So as we reach out to CROs, I've got a lot of comfort that as I reach out to their area that they're doing every day, all day, that they're going to do it well. And it's a great sort of plug and play approach that you can use to discovery. You can scale it up, you can scale it back, just depending on your circumstances. And there's been, as I say, a lot of development in this space over the last 10, 15 years.
Thanks, Chris. So Hamel, you're next up. I mean, you are on the external innovation side, as they were on the CRO side. What do you think collaborators should be thinking about when outsourcing in order to make it a more productive relationship? What do you think they should be thinking about?
Well, yeah, I mean, working in the drug discovery space with external collaborators for so many years, one thing that we have understood is that each project is actually different from the other. So every project has their own priorities, every project has their own set of goals, and the collaborators' expectations is also accordingly. So what we think is, I mean, if you want to make a collaboration really productive, the most important thing is to understand right at the start of the project what the project priorities are or what the project goals are. And if that is very clear, made very clear between the two teams or the two partners who are entering into collaborations, then things become a lot more easy. I mean, then both the parties work in a single alignment and they are able to understand each other. So that's more important about goal settings right at the start of the project, and that should be done very, I mean, meticulously. Otherwise, it consumes a lot of time and efforts explaining each other about the actions one took for the work they were allocated to. So in my opinion, goal settings is something that should happen right at the start of the project to ensure that the collaboration is more productive. The second important thing, in my opinion, is real-time communication. So discussing the things, solve the problems quite efficiently. So I think the communication channel should never dry up. So I rate these two things in terms of increasing and ensuring that the collaboration is successful.
Thanks, Hamel. So I'm going to go to Karin next, and then Peter.
Yeah, so much has already been said. Really starts with investing in a relationship that is a real partnership, equal footing, and that means having clear goals together and having open communication, which also means listening, because your partner can bring a lot. And then building, starting where the capabilities come together, but then also building together a vision, which is win-win. And I think that the aspect of thinking that everything will be just great from the beginning, that is probably the best recipe to never get to great and perhaps, stay at good. So really accepting that we might come from different experiences, setting goals together, but we openly talk together and are committed to build something great, not just good. That I think has been the recipe of success, and very fortunate with our partners, as you know, Sunil.
Thanks, Karin. Peter, same question to you. I mean, you have a lot of experience. You headed up external innovation for Sanofi as well, and plus now you use partners. So same question.
Yeah, I think a lot has been said already. Personal relationships and good communication is very important. And also, of course, when you're outsourcing a major part of your work, that you as a scientist, whether you're in a big company or a small company, is absolutely your baby and is so important. You really, really want the people who are doing that work for you to be as excited about it as you are and as dedicated as you are. And here I'm going to say another controversial thing, which is a bit of a theme in terms of big and small. So I have noticed that obviously as time has gone on, there have been some very, very large CROs created who have acquired and grown and grown and grown. And I just think that in some of our experiences recently, that possibly the same thing can happen to CROs as happens to big pharma, in that when you get too big, you start to lose that personal communication, you start to lose that excitement with people. And so I think that's a criteria for selecting partners. And you can actually tell straight away when you have your first conversation with a potential partner, you can tell around the table the feeling, and that feeling often more than the price is the thing that makes you enter into a partnership with someone.
Thanks for that, Peter. My next question is going to go, there are quite a few questions coming through around some slightly more technical aspects of the discovery process, but I'm going to bring up just one layer. And we all know around Lipinski's rules, and it's been there a long time. And I'm interested to know, do we still feel Lipinski's rules still hold today? And is it still worth understanding, looking at the screening libraries against these, screening our compounds against these before we embark on lead optimization type projects? Peter, I'm going to go to you first.
Great, because this is a great question. Back at Sanofi, I was known as a complete zealot for Lipinski's rules and lead likeness and good physicochemical properties. And it's always good for an argument with a chemist in particular, because people tend to gravitate to two camps, either zealots like me.
I love the argument style of it. So if anyone wants to argue with Peter on the panel,
yeah, I'm ready for you. So yeah, people go into two camps, and a lot of people say, "Oh, it's nonsense. You can make a drug with MEK 600. Look, it's been done." But my feeling is there's so much empirical data showing that physicochemical properties are vital. They predict success in the clinic, and we all want to get drugs on the market. It's hard enough without going down. Without making it more difficult for yourself by going into areas of chemical space that we know are much harder. So if you're given the choice, clearly if you don't have the choice, it's different. But if you're given the choice, I would always gravitate to something that is well within Lipinski's rules, and more actually into lead-like space, because I was brought up with people like Paul Leeson and Tudor Apriya at AstraZeneca, who really pushed the message that it's not just Lipinski, it's lead-like, because when you optimize, you invariably add molecular weight, and so you end up straying too far away from Lipinski. So it's a very powerful, still today, and that was 20 years ago, it's a very powerful methodology for being more successful. So I think it's incredibly important. Just because the original paper was back in the late 90s doesn't mean that it's not something that people should be following right now. And that's the problem, not problem, but that is a potential big downside of PROTACs, of course. So that's a massive thing for them.
Does anyone want to disagree with Peter? Does everyone have a differing opinion here?
No, I just wanted to add, I have kind of like Peter, physicochemical properties are extremely important, and they carry through, and if they don't practice before and in development. At the same time, it's really important to take, and I think Lipinski, as it was also meant in the context, and as we are working through across so many different disease areas, but also different routes of delivery.
Oh, she's frozen. It's a good position, but she's frozen. Give her a second to come back. Chris, what's your opinion on Lipinski? Do you feel that the rules still hold today?
Yeah, I mean, the word rule is bizarre, because I think when Lipinski put this out, I think his intention wasn't as a rule. It was just after analysis of of some properties related to solubility. So if you're going for an oral drug, you do have the barrier of dissolution and absorption to get through. But from a drug design perspective, what I do know is it's easier to make a molecule bigger than it is to make a molecule smaller through the optimization process, which awfully involves increasing potency. It's like skiing downhill versus walking uphill with your snowboard or whatever. It's those are the two different directions to go. That's why I fancy fragment-based drug discovery, because I feel like it's you're drawing your own picture from a blank canvas. However, to explore biology, sometimes you do need tools, and you need probes, and you need things that sort of stick to a protein. And sometimes those can be very undesirable from an oral delivery drug discovery perspective, but they may actually be incredibly valuable from a biology exploration perspective, which could be the first foundation block in a program moving forward. So I think it depends a lot on what your objectives are, and what the purpose is of what you're looking for. And you know, this is why it's not I love to step away from the word rules and just think about, you know, what are you trying to do and what's the best fit for the situation you're in.
Thanks for that. So I've got a question here from David. Well, Karin is probably coming back online, going on and off. Where will David Whitty is, where will MedChem go over the next 10 years, and what will accelerate drug discovery the most? You know, we're all trying to speed up timelines, and maybe the digitization technologies that have been used, maybe some things that we've, like these panels and sessions here today, to improve and keep that community. What do we think will speed up drug discovery the most in the next 10 years? So go with Peter, Hemel, Chris, and we'll go Karin last when she comes back online. What do you think will speed up what we do?
I think two things come to mind. Firstly, of course, the use of machine learning and AI is still probably in its infancy. It's a data-driven business, and so if we can get more and more data into a state where we can look at AI patterns in decision making, then I think that can really make an impact. And only a few companies are really, we mentioned a few already, only a few are really doing that properly so far. So I'm looking forward to seeing how that can really impact. The other thing that I was going to say was it's usually not chemistry that's the bottleneck in drug discovery and even drug development. It's the biology. So I think we need to get better at validating what we're doing, making sure that the molecules and approaches that we have are truly translatable to humans. I think that the big gift there is genetics. And so I think we need to do more and more and more linking our new drug targets and mechanisms to what happens in human via genetics. So I think those two things are going to speed things up.
It's screening technologies and technologies that can mimic more sort of the human body rather than the screening today may be too far removed from a real life.
Yeah, sure, like organoids and things like that. Yeah.
Yeah, 3D. So Chris, if Hemel, maybe you want to make a point, and then I'll ask the last question to Karin. I'll ask her to recap on her earlier point and ask her the current question, which she may not have heard. So Chris.
Yeah. I mean, I was going to kick it back the same way Peter did, to the sort of the biology side of things, that you know, a deep understanding and development of biology and translation, etc. Then you know, drug discovery, you know, way back when, before the invention of the in vitro screening cascade system, a lot of it was based on organ tissues and organ baths, and so your compounds need to be soluble. The phenotypic effect was right there in front of you. You know, how can we incorporate some of that approach, but then using more technology-based approaches to, you know, using human-based systems through the discovery process earlier rather than later. But yeah, I think a lot of failures are really to do with either lack of efficacy or resistance pathways that develop. And this is why earlier I was talking about combinations and bringing combinations into the thought stream earlier on in the process rather than later.
Thanks, Chris.
Yeah, I also tend to agree with Peter and Chris that machine learning is something that will be definitely, AI will be something fascinating to look forward in future. I mean, in terms of chemistry, the most challenging aspect is the route scouting. I mean, having a right route for the molecule determines the success right at the start of your reaction, right? So, till date, we have been using traditional database like Skyfinder or Reaxys, which just goes through what is there inside the patents and gives the hits. So it depends on a personal search. I mean, it depends on a person's quality to search about his or her problem. But if we add a machine learning component to that, where the robotics can come into place and help the development of that route in a better manner, and I think problem solving or having better routes become possible right at the first instance. In the same way, I'm looking forward for some platforms which are able to review our spectra and just help us to confirm what if we have isolated the right compounds. Many times we struggle with understanding the byproducts. So if there are certain platforms which are able to contribute on that with the help of machine learning, then that can also definitely increase the productivity of chemistry. Yeah.
Thanks, Emma. Great answer. So, Karin, if you just, we're going to finish off with your answer, so no pressure. But if you just wanted to finish the point you were making earlier on Lipinski, and just to wrap the session up, to say how we can improve the speed of drug discovery, what's going to have the biggest impact over the next 10 years was the question from David.
Thanks, Sunil. And I wondered if my computer censored me before, but really just adding again, physical chemical properties are really important to consider. It has to be done in the context of what one wants to accomplish, and it's one thing on oral drugs going systemically distributed. We are also thinking much more about having local deliveries, or really just restricted to one tissue, and then there are other properties to be considered, but the properties have always to be considered. It's just in that context. And of course, also when we think about more long-acting drugs, and that was really my point there. Very much in agreement with Peter, just saying that there is a broad scope we need to think about, and so Lipinski extended to the purpose. But very much in agreement, where the technology is going, what are some the biggest hurdles to accelerate drug discovery? One aspect is clearly the targets, what are we going after, and how we get to the high-hanging fruit, which I think we have made a lot of progress over the years and continue to do that, with really drugging very difficult, impressive protein-protein interactions, for example, bringing complexes together. But at the same time, I also think that the translational systems, what are the diseases we haven't really gone after so much, are the ones where actually even not only not having a translational system and discovery to work on, but some of the neurodegenerative diseases where if we go into the clinic, it takes so long to see if we have had an effect. And that makes it very difficult, if the success is years away. And so really working on understanding the underlying biology and pathways of the diseases. And that's not only a biological problem, it is, of course, a biological medical problem, but chemistry has been so powerful in helping with understanding biology, really thinking about the atomic resolution of the disease. And so as chemists, we can contribute there a lot. We call it chemical biology, but I call it good chemistry, bringing in the molecular thinking at the atomic resolution to all aspects of drug discovery, partnering with biology and the physicians to really understand disease better and and how we can actually monitor it in the clinic. That's the biggest failure rate we have, and that's what we need to crack so that we get into the neurodegenerative diseases we would like to, and also get to cures rather than just treatments.
Thank you for that, Karin. That's a great way to finish off the panel. I just want to reflect slightly. It's been a turbulent year for most people. We've had to deal with new things, challenges apart from the science. I wanted to, on behalf of Prashant and I wanted to thank all the panelists that appeared on our ChaiTimes over the year, spent time coming on, all our collaborators and partners, wherever they are in the world. Everyone's been really supportive, going the extra mile. There's been a lot of understanding on both sides. We've all tried to work this through together. And I just want to just to really appreciate everybody that's been a part of our ecosystem has helped or supported us in a small or large or meaningful way, all the understanding that everyone's showing to each other, everyone's being considerate, compassionate. And I think one thing the pandemic has shown us is that we are really compassionate human beings, and we understand. And great thanks to everybody. I just wanted to finish off our last season 2 Chai Time on that positive note. We are really looking forward to next year. There will be some more Chai Times next year, pandemic or not, and we look forward to some more great panel discussions like the one we've had today. Thank you very much for your time. Thanks for listening. We'll speak soon.
Thanks, Sunil. Thanks, everyone. Happy holidays.
Thanks, Sunil. Bye-bye, everyone.
Bye, everyone.